So good morning and good evening, everyone. My name is Juliana, and today I'll be presenting my dissertation that was entitled Unraveling the Global Virome of Bees Using a Small RNA-Based Approach. Bees are insects from Hymenoptera order that are found in all continents except for Antarctic, and more than 25,000 species have been described until now. They can have either solitary or social behaviors, and they play an extremely important role in pollination. They are fundamental for the maintenance of agricultural activity in a global perspective, and they also help to maintain native plants. Also, some species are able to produce honey and other derived products. And all of these reasons had led the bees to be largely cultivated in the last centuries. Because of this cultivation, the beekeepers have noticed on the beginning of the 2000s that the bees were starting to disappear from the hives. They were leaving behind food stocks, their brood, and they were simply disappearing. And they called this disorder as colony collapse disorder at the time. And several reasons have been associated with this disorder. For example, the use of pesticides, the radiation, climate changes, and also the incidence of parasites and diseases. And I will be focusing on parasites and diseases on my dissertation. So there are several parasites and pathogens that affect bees. Mites and viruses are the ones that are mostly related with the CCD. And more specifically, a mite that's called Varroa destructor and a virus that's called the deformed wing virus. And these two pathogens are extremely related because this mite is the vector of the deformed wing virus and all the other viruses as well. And here you can see a picture of the mite feeding from the bee. And this is when it will inject the viruses such as the deformed wing virus. And in this case, in the case of this virus, they can cause this deformed wing, as its name says, and it eventually leads to the death of the bee because it becomes unavailable to fly, so it dies eventually. And the symptoms of the CCD, they are usually associated with co-infection of bees with different pathogens and not only one pathogen. For example, the association of mites and viruses. 24 viruses have already been described infecting bees from Apis genus, but little is known about viruses that can infect other bees. And there is still a lack of information about the circulation of these viruses globally. One way we have to study viruses today is through the metagenomics, which studies the collection of microorganisms that are present in a sample. And basically, we have individual samples or pooled samples that will be extracted for their genetic material. That can be DNA or RNA, for example. And among the RNA, there are several classes that can be chosen to study. For example, long RNAs and small RNAs as well. And after you decide which kind of molecule you want to analyze, we go to the next generation sequencing, or NGS, that will produce several reads that needs to be pre-processed for the removal of sequencing adapters and reads that don't have enough quality to be analyzed. So this is the pre-processing step. And after the pre-processing, we go to the contigs assembly. So several reads are put in together into a large sequence that's called contig. And finally, we go to the bioinformatic analysis, which will associate each of the contigs assembled to an organism. And in this step, we have a problem that is known as the dark matter of the metagenomics, which are the sequences that don't have any similarity with known organisms. So these sequences, they are unknown, and this is a huge problem today. And one way of dealing with this problem when it comes to viral sequences, new viral sequences, is through the analysis of small RNA libraries. Because when we use small RNA libraries to assemble viral contigs, we can use analysis independent of sequence similarity to find potential viral sequences. For example, when we see here this kind of profile, the small interfering RNA profile, which consists of the accumulation of small RNAs that range from 20 to 23 nucleotides. In the case of bees, is 22 nucleotides. When we see this kind of profile, we are seeing an evidence that that sequence comes from a virus that was essentially replicating in the host and causing an immune response on the host. Also, we are able to differentiate endogenous viral elements from exogenous viral viruses when we look at the small RNA profile. Because when the virus is an exogenous one, we see the small interfering RNA pattern. And when we see an endogenous viral element, we see this kind of profile, that is the pi-RNA profile, in which the accumulation of small RNAs occurs in the sizes of 24 to 30. Also, when we compare the viral contigs assembled using small RNA libraries versus the long RNA libraries, we see normally that these viral contigs have larger sizes, larger number of contigs are assembled, and they have also a larger coverage. And this all happens because in invertebrates, the small RNA portion is enriched for viral sequences. This is thanks to the RNA interference pathway, which consists of 3 other pathways that are called microRNA, piRNA, and small interfering RNAs. And this last one is the main antiviral pathway in invertebrates. So we are going to focus on this one. When a virus is infecting a host cell, it starts at some point replicating. And during this replication, it produces a double-stranded RNA that will be recognized by the cell as a threat because it normally doesn't occur in the host cell. And this recognition will induce the small interfering RNA pathway that starts with the recognition of this double-stranded RNA and the cleavage of this double-stranded RNA in several small pieces. These small pieces will be used by a protein complex to detect sequences that are complementary to those small pieces of RNA. And through this recognition, this RNA will be cleavage, will be cleaved. And this process leads to the production of small fragments of RNA that goes from 20 to 23 nucleotides, as I said before. And when we analyze the profile, the small RNA profile that comes from the sequence, this is what we see. So here we have the number of small RNAs in the plus strand and the negative strand. And here we have the sizes of the small RNAs. So as I said before, in the bees, when we have this kind of response, we have the accumulation of 22 nucleotides RNAs. So we see this accumulation in both strands. And this is an evidence that we are actually looking to a virus that is replicating in the host cell and generating an immune response from the host. So having that said, my general objective was to unravel the global B virome through the analysis of public libraries of small RNAs. And among my specific objectives are identify and characterize through sequence similarity analysis viral sequences that are present in the public libraries, analyze the profile of small RNAs from all assembled viral contigs so that it's possible to infer the host's immune response and differentiate exogenous viruses from potential endogenous viral elements, as I explained before. Also perform independent sequence similarity analysis using small RNA profiling to find potential virus sequences among the highly divergent contigs, which are the contigs that don't have any similarity with known sequences. Relate the geographic location of the analyzed bees with the viral species found, and finally study the phylogenetic relationships of the new viruses found in the analysis. And regarding my methodology, it starts with downloading raw libs from the SRA, which is a database from NCBI. And then we do that pre-processing that I showed before, in which we do the quality control and we exclude all the adapter sequences. And after that, we do the alignment against reference genomes of bees and known bacteria. This way we are excluding all of the sequences that comes from the bees and the ones that comes from bacteria as well. And using unmapped reads, we do a de novo assembly. We use two assemblers, Spades and Velvet. And after the contigs are assembled, we make a sequence similarity analysis, first at a nucleotidic level, and after that at amino acidic level, both using the tool BLAST. After that, we go to a manual curation that consists first of small RNA profile analysis from viral and contigs no hit, which are the ones that don't have similarity to anything. After that, we make the open reading frame analysis and we search for conserved domains on these contigs, followed by a clustering of these viral contigs and no hits that were manual curated. And we make also a co-occurrence analysis. And finally, the phylogenetic analysis of the new viruses that were found. So this is my first result. Here you can see all the public libraries that were selected. We had in total 131 libraries, public libraries. And here we can see each of these colors are different bee species. And here you can see the number of each bee that was collected and where they were collected from. And we can see here that most samples available are from Apis mellifera, followed by Bombus terrestris, and a few species that are not very well studied. We have only 1 or 2 samples. And when we analyze these samples, what I'm showing here is the total number, sorry, not the total number, the percentage of contigs from each of these classes that were assembled for each of these libraries. So here are the bee libraries, and they are separated by each color is one bee species. And each of these categories here are, in purple, we have the no hits contigs. In blue, we have the non-viral contigs assembled, which are contigs that had similarity with something, but it wasn't viral. and the ones that we are really interested, which are the viral contigs. We're also interested in the no hits because here we have probable new viruses. And we have a cutoff of 199 nucleotides for the assembled contigs. So only 112 libraries had assembled contigs that were greater than this cutoff. And here we are showing only those 112 libraries. And these bars are showing the percentage of contigs from each of these categories for each of the small RNA libraries. So what we are going to analyze from now on are the viral contigs and the no-hit contigs. So the first step is to manually curate these contigs. So we had 343 viral contigs in total in all of the 112 libraries. And the manual curation consists of 3 steps. The first one is a curation of the top 5 hits from BLAST, followed by the open reading frame structure and the analysis of the small RNA profile to see if these viral contigs, they actually have those small interfering RNA profiles that we expect to find for viruses. And after this manual curation, we went to 317 viral contigs, and we did the same for the no hits that were from 215 to 34 after manual curation. But in the case of the no hits, we only have 2 steps of manual curation because they don't have similarity with sequences on BLAST. And after the manual curation, we selected all of these contigs and we clustered them by similarity using the CD hit, which resulted in 146 clusters. These clusters, they are showed here in this heat map. Each of these each of these columns are one representative contig from each of those 146 clusters. So we have here 146 representative contigs. And here we have all the 131 libraries, small RNA libraries. And we clustered these representative contigs by the distribution of small RNAs between 20 and 22 nucleotides. And so they clustered mainly by viral species. So here I manually separated them by viral species. And we had 14 clusters in total. And here we are seeing each of these colored dots are showing where these representative contigs are found in all of the libraries that were analyzed. And here we have a brief summary of which viruses are related to each cluster. So for example, Ube particulite virus 34 is found on clusters 1 and 4. Apis rhabdovirus 1 is found on clusters 2 and 5, and so on. And interestingly, the clusters 7, 12, and 13, they contained contigs that either presented similarity with viruses at amino acidic level or contigs without any similarity to known sequences, which are the no hits. So we call the viruses from these clusters as new putative viruses. And here we have a summary that shows which viruses, according to the heat map, which viruses were found in each bee species. So only 4 bee species presented viral contigs in our analysis. Here we are showing the name of these viruses, the viral species, And here is the similarity with the best hit from blast for each of these viruses. And interestingly, here, each color of these lines represents a different type of genomic organization. And our approach was capable of detecting viruses with different types of genomic organization, as you can see, as long as there is production of double-stranded RNA at some point of their viral replication. Another interesting thing was that we had the one library of this bee, Megachili rotundata. It's a solitary bee, not very well studied yet. And even though we only had one library to analyze, we found 4 different viruses in this bee. And 2 of them seems to be new viruses, as you can see here. So this shows that there's still a lot to learn about those not so well studied bee species. While talking now about the phylogenetic analysis of the 3 putative new viruses. First, the virus from cluster 7, it grouped with viruses from Solemoviridae family, so we call it new Solemoviridae. On cluster 12, our putative new virus grouped with other viruses that weren't classified in family level, but they were from Picornaviralis order. So we called it new picornavirus. And finally, for cluster 13, we had the virus was grouping with viruses from Helviridae family. So we called it new Hel virus. And the general idea that we can take from the phylogenetic analysis is that the use of small RNA libraries, it limits the size of assembled viral contigs. So as a consequence, the size of our DRPs is limited, and this can influence the quality of phylogenetic analysis because we use the DRP to make our phylogenetic analysis. So this is one of the limitations of our technique. And here I'm showing the small RNA profiles from the viruses that were detected in the analysis. So each line here represents one of the 14 clusters that I showed in the heat map. This is just to show you that most of the viruses that we found, they have this classical small interfering RNA profile. And one interesting thing is that on cluster 7, that is related to the new Solemoviridae, this viral family, it infects mostly plants, and here it was found infecting Bombus terrestris because we see the profile that shows that the virus is actually replicating in the bee. So possibly this bee species could be a vector of this virus for plants. And another interesting thing is that on clusters 8 and 10, we didn't find that classical small interfering RNA profile. And these 2 viruses are from the order Picornaviralis, in which we have already, it has been already described some viruses of this order are capable of inhibiting the small interfering RNA pathway. It's a way of evading the immune response from the host. So we believe this may be happening here, but there's still a lot to investigate. And finally, the last 3 clusters also showing the small interfering RNA profile, the classical profile. And finally, the distribution of viral families among the countries where the samples were taken from. So as you can see, the most spread family, viral family was Rhabdoviridae, followed by Ethylviridae. And unfortunately, most of the viruses that we detected are yet not classified. So this shows that there's a lot to be discovered in bees. It's a very interesting area and it should be more studied. And that's all I had to say for today. Thank you very much for the attention, and I will be open for questions and discussions. And yeah, that's it. Thank you very much.